For inviting me and also the great introduction. So hello everyone, I'm Ruishan. So today I'm going to talk about many of my two projects to demonstrate how machine learning can be used to help clinical trials and precision medicine. So you can just interrupt me at any time with questions. Okay, so I'll give a very short introduction. So if you think about the streamline for medicine, so it may have two stages. The first is drug development, where people develop new drug and new proteins. And then those new drugs will be sent to clinical trials to test for the efficacy, toxicity, etc. So after the drugs are approved, then they will be on markets and the clinicians or physicians would give it to the patients. So if you think about, say, how AI or machine learning can help in this streamline. So I'd say we already have a great success in the very first stage. So for the drug development, for example, we will have very well-defined machine learning tasks to design the molecules, to predict the protein structures, and also we can use models, machine learning models, to do drug screening, like say to predict the toxicity and like those different properties of the drug. So today, my talk would mainly focus at those later two stages. So after the drugs are developed, how can we use like the modern AI and machine learning to help the clinical trials design and also to better usage, like in the real practice? So more specifically, I'll mainly talk about our projects. First, the Trial Pathfinder, like Ahmed also mentioned, how to develop algorithms to optimize the enrollment criteria for clinical trials, and also say how to generate new hypotheses for precision oncology. Okay, let's get started. So in the first part, I'll mainly talk about the Trial Pathfinder, like our first projects, say develop machine learning and also use real world data to help enrollment criteria design for clinical trials. Yeah, let me give you more background about this. So modern clinical trials are the bottleneck of medicine. So if you look at this figure where we show this, so from hundreds of new drugs you develop, only very few of them can pass through the clinical trials. So this process is very slow, expensive. And you can see here, the average cost of phase 3 clinical trial is around $20 million. So it's very expensive and costly. So one critical barrier for the clinical trial is the strict eligibility in their enrollment process. So what does this mean? So here I just show some statistics about 10 non-small cell lung cancer trials we analyzed. So within the pool of patients who are willing to participate, only 30% of them can pass through, are eligible to get into the clinical trials. So that is the pharma companies, like they usually set very restrictive enrollment criteria. And also only the patients who satisfy all the rules can be enrolled in the clinical trials. So for example, they usually would tend to have relatively younger and relatively healthier patients to be enrolled. And from the 10 lung cancer trials we analyzed, only 30% of them can be enrolled. So as a result of the very restrictive eligibility and the very low rates of enrollment, 86% of the clinical trials fail to complete recruitment within targeted time. And also there's a generalization problem. So the trial cohort doesn't represent real population. So if you think about this, if you always just to test your drug on relatively healthier patients, you do not really know how they would perform on sicker or older cohorts. As a result, there's a very strong need and advocates to relax the enrollment criteria for clinical trials in both academia and industry. So NCI's clinical trials program has concluded that the eligibility criteria for all cancer clinical trials should be simplified. So this comes to our project. We propose an algorithm called Trial Pathfinder, where we can evaluate and optimize the eligibility criteria for oncology cancer trials using real-world data and AI. And as a very quick result, we're able to double the eligible patients from 30% to over 60% without harm the performance of the drug. And also, we are very excited, like our algorithm is currently piloted by Roche, one of the world's largest biotech company, to guide their cancer trial design. So the screenshot is the website we developed, the internal website we developed with Roche. Like they can help the clinicians to design their new clinical trials. And also we are very excited. Trial Pathfinder was recognized as a top data science innovation by the pharma industry in the past few years. Okay, yeah, so I'll dive into it and give you more background and how we design the algorithm and how it actually works. So first, I want to give you more sense about, say, currently, what is the current stage of how people design eligibility criteria in pharma industry. So this is in our collaboration with Roche, and this is a real working protocol. Like they are currently designing this for a new drug. And this is so how we collaborate and how they actually works in real time is, so this is a screenshot of a Google Doc. Just as how you collaborate, so like with your collaborators, and you have a very long Google Doc and to state different rules for eligibility criteria. So here I just took a screenshot of a few of them, of lab rules. So how it goes is they usually have two steps. The first one is they usually duplicate many rules from previous trials. And then they will have a team of clinicians to review and to decide whether to edit or add or remove rules. So for example, this is like a real example. And for example, they will have a team of clinicians and they will decide, oh, before the threshold for one rule for ANC is 1,500. And one of the clinicians just commented and to say, oh, can we change it to 1,000? And other clinicians will reply to say, oh, I feel maybe not so safe, and some other things, oh, probably not. So you can see this whole process is pretty anecdotal. So that is to say, mostly depends on the clinicians, their own feeling and their own experience. So if you look at the real world, if you have any experience working with the real world protocols for clinical trials, we can say they have very diverse threshold and rules. So that is a reason, because you do not really have a evidence-based or data-based or any tool to help you to decide, but you only make the decisions based on the clinicians, their own experience. So why it is so difficult to provide a more general guidance or like evidence tools for it? That is, if you think about the clinical trials design process, one key question is, you would never know what happened to the patients who have been excluded by the clinical trials. So for example, if you imagine you were a clinician who designed a clinical trial, and as a clinical trial carry out and one drug get approved, in the future, you want to design a new protocol and you suddenly want to say, oh, can I relax it? But then you realize that you never know what happened to the patients who outside your rule, like you designed before. So you do not have any support to say, oh, whether it's good to relax it or not. So that comes to our projects. So our key idea is to say, for the real clinical trials, you do not really know what happened to the patients who have been excluded because they never entered the clinical trial. But after the drugs are on markets, because all the patients need to get medication, like old patients and sick patients, they need to get medications. So on the real world data, like after the drugs are on markets, you can see diverse patients who take that drug, and then you can go back to answer the question, say, oh, for all those different rules, how do they really affect the drug's outcome? Yeah, so that is the key idea. So based on that, we propose our framework Trial Pathfinder, where it composed of two main steps. First, we try to evaluate, simulate clinical trials from real world data. And the second step, we make use of those clinical trials in silico to evaluate and optimize eligibility criteria. Okay, so in the very first step, we want to emulate clinical trials from real world data. So that is, you will have a medical records for the patients. And on the medical records, you can know what drugs the patients take, like the health condition and what the outcomes is. And you want to, and here we want to design the process to say, oh, we just perform as if the patient is in one clinical trial. And if you think about what are the necessary steps for it, and here is our answer. The first, we try to encode the eligibility criteria for the different protocols. So those are all written in English statements or different language statements. And we need to transfer it into machine readable rules. And the second, because in order to emulate clinical trials, we need to generate two groups, like the treated group and control group in the clinical trials. So how we do it is we have the medical records for all the patients, and say suppose we want to generate a clinical trials who test the drug A, the performance of drug A versus drug B, and then we just identify the patients who took drug A as a treatment arm, and patients who took drug B as a control arm. And also we want to follow the process of the clinical trials, and we need to check all the patients have to satisfy the enrollment criteria for the clinical trials. Yeah, before, well, we can get the performance, or like say, can emulate the clinical trials. One critical part is we have to emulate the random assignment in the clinical trial. So the one largest difference between the real clinical trials and real world data is in the clinical trials, all the patients, they will be randomly assigned in different arms, but then in the real world data, there could be a lot of selection bias. So for example, the clinicians might tend to give sicker patients drug A more frequently. So in order to do that, we have to emulate those random assignments. So that is, we have to match the two distributions for the treatment arm and control arm. So there are different ways to do it. So the two large category is, first, you can do the weighting. So basically you can do some propensity score-based weighting. And because this is like a very high-dimensional data, you will have to build it on top of some machine learning tools. And also you can directly do the matching. So for example, you can using optimal transport to match the two distributions, and we'll have some like advanced tools proposed to do that matching. So then at this step, you already have two groups, and also you have matching their distributions, and then you can do the analysis. And here I just give you one example, like one metric the clinical trials used. So you can do the survival analysis on those two groups. You compare the survival performance in the treatment group and control group. So here is the survival curve. So I think here maybe people are already familiar with the survival analysis, but if not, I'll give you like a very quick introduction. So here in this graph, in this figure, the x-axis is the enrollment time. So from the patients enroll and also like as you follow up as time pass by. And the y-axis is the overall survival. So that is the percentage of the patients who are alive. So that is in the beginning when all people get enrolled as like x-axis start like at 0, all patients alive, so it's 100% survival. So as time passed by, the patients died, passed away, and the curve decreases. So that is so your survival percentage decreases. So in this case example, so the treatment group, the patients in treatment arm just passed away slower than the patients in the control arm. So that is to say your new drug, the drug in treatment arm is perform better than patients in the control arm. So we will have a metric for this. We call it hazard ratio. And here, the hazard ratio less than 1 means the drug in treated group performs better than the drug in control group. So if you do not have background in the survival analysis, you just need to keep in mind the hazard ratio less than 1 means your new drug, the treated drug, is better. Okay, so after you have this whole process, like rule encoding, treatment groups generation, random assignment, and also follow up different analysis, for example, survival analysis, you're able to emulate clinical trials from real world data, from your medical records. So just to remember, our main idea is to help design the eligibility rule for clinical trials. So the next step is we want to make use of the first streamline to help evaluate and optimize eligibility criteria. So here I just give you 2 examples. Say, for example, here we are able to answer the question. So what about I only have 2 rules, like rule 1 and rule 2 in the enrollments? How many patients I end up with? And what is the hazard ratio? So here I just use hazard ratio as one metric to demonstrate like the drug's efficacy. And also, say then you can also try to look at, say, if I only have 1 rule, like rule 2 in the criteria, how many patients I end up with, and how would that affect the drug efficacy? And so in light of that, we can emulate millions of trials with different eligibility criteria to look at, so how many patients I will have and how would that affect the performance. So you can see like our ideal way or like what we care about is we want to recruit as many patients as possible while we do not want to harm the performance. And we also propose a principled way, like a metric, to evaluate the individual enrollment criteria. So we propose this a very principled machinery way, like a Shapley value for hazard ratio. So here, the Shapley value, if you look at this definition, like you just need to focus at the right part, is an average contribution of hazard ratio of ith criteria in combination of the other subset versus minus the hazard ratio of the other subset. So basically it is the like what are the contribution of ith criteria with some other subset of criteria. And the Shapley value is a principal way you can get an average contribution of ith criteria in combination of different other subsets of criteria. Yeah, so to this point, you have the idea, say how we can emulate clinical trials from the real-world data from medical records. And further, you have the tool to evaluate your enrollment criteria in clinical trials. And we implement our framework. Oh, yes?
Yeah.
Is there a question? Yes.
So like, how do you come up with the set of criteria that you are comparing? Because like the criteria might be like ranges of variables, like it can be like continuous, like do you start with like a set of discrete criteria first, and how do you select these?
Yeah, that's a very good question. So like our project is to look back all the old clinical trials, to look back on their criteria to evaluate how well their decision about criteria. So basically here the Shapley value is the very first thing is like you look at all the sets criteria the clinical trials have made. So basically for one clinical trial, you look at their protocol and you extract their criteria. And here, like the other criteria are like, say, what in real time, what are the other criteria used in companion with that criteria. And also like there is a very good question about say, because a lot of thresholds are continuous. So how we do that? So I'll show in the I think like in a few slides to say you can actually change different thresholds to say how does it affect the Shapley value in companion of other criteria. Yes. Okay, yeah. And so we implement our framework on a real-world cancer database, Flatiron cancer database. So the Flatiron network has more than 2 million patients where we have their medical records. So you can extract the patient's treatment information, follow-up information, health conditions from their medical records. Yeah, here I'll give one example. So just exactly as Ahmed asked, because for in real time, like a lot of rules, especially for lab rules, they will have different thresholds be used. So how can we use Shapley value to help decide what threshold we can use? So here I give one example of 10 lung cancer trials. And so they are all targeting like non-small cell lung cancer. And you can see here, we extract the threshold they used for bilirubin. So bilirubin is like a standard metric for liver function. And you can see there are two main versions for the bilirubin threshold used in literature. And also just if you recap what we introduced in the beginning, how the clinicians decide that threshold is very anecdotal, like based on, so that it mostly depends on like different groups of clinicians, their own experience. And what we can do is say we can actually test what if we leave all other criteria as what they are, and we just change the threshold for bilirubin, how will that affect the Shapley value for the bilirubin rule? So here the x-axis is the threshold of bilirubin, and if you increase the threshold, that rule is less restrictive. Yeah, so you can see actually for like all the 10 clinical trials, it is pretty safe to relax the bilirubin to the least restrictive version. Yeah, and also we can give a very quick recommendation. So we can just give the clinicians one recommendation of all the criteria with negative Shapley value. That is all the rules, if you include them, you tends to reduces your hazards. So we call it data-driven criteria. So here you can see we compare the number of patients and also the hazard ratio between the original trial criteria and our data-driven criteria. So you can see here, the data-driven criteria can double the number of eligible patients while even reduces the hazards for the drug. Yeah, just I want to give a very quick summary here. So our Trial Pathfinder, we propose a data-driven approach to make clinical trials more inclusive. So apart from what I introduced earlier, we also validated it on the clinical trials data and also independent cohorts. And apart from lung cancer, we also see similar results on other cancer types, including colorectal cancer, breast, and melanoma. So we would expect it's like the Trial Pathfinder to expand to other diseases also outside of cancer and also to different database formats. Yeah, and yeah, I can make a short pause here to see if people have questions in our first project. Yes, Alexander.
Yes. It's like how do you draw a line here, right? Because like basically you're kind of extrapolating from your emulation of like past trials to now criteria for a novel drug, which might have like some different properties, right? So where do you draw that line of like that is like a class of drug where these previous approved drugs or trials are representative, or where you maybe cannot do that as simply as it might have worked like for some of these cancer examples here?
Oh, that's a very good question. So our framework, what we do is we give clinicians a tool to evaluate all the past clinical trials. So basically, like say a key question the clinicians have is after they make the decisions before they do not really know how well their decision end up with. So for example, if you look at the screenshot I take here, like the tool we developed together with Genentech or with Roche is say if a clinician like make a new drug in the future, like we can recommend them with a drug in the same category or like same mechanism, and they also can freely choose the drugs they think can be a reference to the drug they have. And also, say for clinical trials, actually not all the trials are making for the new drug. So basically, like a lot of them are trying to do like off-label, like a different, say, so I think like more than half of the trials are actually on the old drugs. So those are pretty simple. So we can just return all the old drugs for this, all the old trials for that drug, to say how different trials, how different rules would perform. But for new drugs, say if it's like entirely new, because nothing would happen. Because like to the best people would know is like you can give them the guidance of drugs in the same category and also say with similar mechanism. And also I want to mention one result we found is actually for one cancer, we do see like say like a lot of lab rules, they consistently are useless across different drugs. So that is, we do see some like common knowledge or common sense for different cancers. So those are pretty safe to provide to the clinicians as well. Great. Is there any other question? Okay, cool. So yeah, I will just conclude on my first parts of the talk, the Trial Pathfinder. So I hope you can get the idea how to use real-world data, the records, to help evaluate the enrollment criteria for the clinical trials. Yeah, so then I'll move on to our second part of the talk. So after the drugs on our markets, how can we generate new hypotheses for precision oncology? So I think like people like joining this meeting already know like what precision medicine is, but so then I'll just quickly go through. So here, like what we refer to is I just cited from the Precision Medicine Initiative. It's a disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So just just like instead of the one drug fits all style, now we want to give patients individualized care based on their different genomics or different environment lifestyle factors. So here, so I want to give you some basic sense of what we analyzed. So from the 8 cancer types we studied, so the current stage for precision oncology is for the patients on cancer therapies in the US, only 15% of them have genome-targeted or genome-informed cancer treatments. So by genome-targeted, we mean that FDA-approved treatments that can target certain mutations. And by genome-informed is from if there's any evidence in literature, people have found that the patients with certain mutations have some targeted drugs. So you can see still like our knowledge is currently is very limited. So only have 15% of patients can benefit from the targeted drugs. And based on that, so comes to our projects, saying similar to the first part, the Trial Pathfinder. Here we want to make use of the real world data, the medical records. So say given the real world data, medical records, you can know what the drugs patients take and also their genomics profile and their outcome. So then you can answer the question saying, given the patients with certain mutation, what be a better drug we can give to them? And as a quick result, like we are able to generate new hypotheses. So we're able to increase by 20% more patients. We have hypotheses on like they can take certain drugs to target their different mutations. And to do that, like we link the Flatiron network I introduced in the first part, like where we have the patient's medical records for all the cancer patients, and link it with the foundation medicine data, where we have the patient's next generation sequencing for more than 500 cancer driver genes. And as a result, after the matching, we have more than 40,000 patients where we have both their mutation profiles and treatment outcomes. So then we're able to do the analysis to say generate hypotheses about say what would be the predict biomarkers for different treatments. Yeah, so to do that, I want to also introduce the interaction model we built because we want to get a principled way to evaluate like the interaction between the gene mutation and treatments. And we also adjusted it by the patient's confounders we extracted from the medical records, such as the age, gender, histology, stage, of all different covariates. And here we build this, so there's a Cox function. I think people here maybe or may not, so I'm not sure like people's background here is, but like here you just need to know, say the hazard ratio. So there's a semi-parametric model. So basically here we want to model the instantaneous death rates of the patients. And here H0, so it's a semi-parametric part. So you only need to focus at the exponential parts, right? So for the exponential part, you have the contribution of mutation alone, like mutation G alone, and treatment alone. And what we care about is the interaction between the gene mutation and the treatment. So here, that is the interaction term. And we call this term as the mutation treatment interaction hazard ratio. And here, the interaction hazard ratio larger than 1, it means if you have that mutation, you're not really recommended to take that drug because the hazard ratio larger than 1 means like you have a worse survival. And we also, because for the medical records, we also need to do left truncation and right censoring, like to control for the immortal time bias and also for the limited follow-up. Yeah, I'll give you one case example to let you interpret, say how what does the interaction hazard ratio mean and how we can use that. So here, I'll show one example of how gene APC, so here APC is a gene name, interacts with immunotherapy in advanced bladder cancer. So here the gene is APC, drug is immunotherapy, and we look at all the patients who have bladder cancer, right? And we do, we run the model and we get the interaction hazard ratio to be significantly less than 1. And that means APC is a positive predictive marker for immunotherapy. So what does this mean? So you can actually view this problem from two aspects. So first, you can look at all the patients who took therapy with bladder cancer, and you compare the performance of patients who have APC mutation versus patients who do not have APC mutation. So here in the survival curve, like the orange curve is all the patients who have APC mutation. You can see they tend to die relatively slower than patients without APC mutation. So that explains your interaction hazard ratio less than 1. That means immunotherapy performs better on the cohorts who have that mutation, right? And you can also look at this problem from another aspect. So you can say you only focus at the patients who have APC mutation, and you compare the performance of immunotherapy versus other drugs. So similarly, you can see here, for all the patients who have APC mutation, like if they have immunotherapy, that tends to pass away like relatively slower than patients on other drugs. So those two together explains what does it mean of interaction hazard ratio be significantly less than 1. So here, APC is a positive predictive biomarker for immunotherapy in bladder cancer. And also, say why I use this case example is because like so here, what we do is all real world analysis. Like so we adjust for different confounders, we build this model. And also, so for bladder cancer, we are able to extract a few clinical trials where they have records for the patient's genomics profile and for one immunotherapy drug. And similarly, we found the interaction hazard ratio in the clinical trials are significantly less than 1. So similarly, so inconsistent with what we found in the real world data. Yeah. So based on that, so we can do this systematically. So we can look at all different cancer types, different treatments, and different mutations. So we do this systematically on the Flatiron matched Foundation Medicine database, and we identified more than 400 mutations that significantly predict the outcome of patients on specific treatments. So here, I visualized a subset of our findings. So if I look at this plot, a different large block indicates different cancer types. For example, the leftmost is advanced non-small cell lung cancer. And here we use, so you can say they have a different gene name and also like different sizes of the gene names. So here, the size corresponds to the percentage of the positive patients given the treatment. And also the color indicates the interaction hazard ratio. So if the color is red, means the hazard ratio larger than 1. So that is that gene is a significant negative predictive biomarker. That is, so you are not really recommended to take that drug if you have that mutation. So for example, if you look at the leftmost or bottom left, say on the category of targeted therapy, you can see there is a EGFR inhibitors block. So that block gives all the a subset of significant predictive biomarkers for EGFR inhibitors for non-small cell lung cancer. So EGFR inhibitors are a class of drugs where like they are already known, like approved by FDA to target EGFR mutations. And you can see from the data, we also found EGFR, like the large blue, to be a positive predictive biomarker for EGFR inhibitors. So just exactly as what we expect. And so here I want to emphasize that, say, we do have a few case examples, as I show, like EGFR is a predictive biomarker for EGFR inhibitors, but most of our findings are hypothesis generation, like say people do not have enough like a large sample size, or like they do not have any reports. So those would be a very good candidates for precision medicine clinical trials to test for. Like those are good candidates, biomarker candidates. Yeah, I'm going to pause a bit here to see if there are any questions for like this pan-cancer analysis for the gene treatment interactions.
I think one question is like, did you need to use like the Cox model with an explicit specification for the interactions? Like if you wanted to use like an arbitrary machine learning model, would that, like, what would change in terms of the rigor of your analysis?
Yeah, that's a very good question. So for this project, so I would like answer this from two points. So the first is actually we have like other or like more complicated machine learning models like random survival forest or different other models to predict the patient's survival based on their mutation profile. So I would say they do have better performance than the Cox model, because again, the Cox model is a second order, like linear model. Here, but I would say like here, why in this project we use that Cox model, because it is more interpretable. So that is to say, use the Cox model, we are able to extract the interaction between individual mutation and the treatments. So basically, that is we can have this very nice predict marker like figure to show to the clinician. Let's say they have like a patients with certain mutations to say whether they are predict biomarkers for different treatments. So basically, it's more for interpretation purpose, and also you will have a very good like confidence check. So basically, it's mainly for the interpretation purpose. So in terms of the prediction power, so we do have like say other models. So that would be like would outperform the Cox model. Yeah.
Yeah, and because the goal is hypothesis generation, not accuracy of prediction, that's why the Cox model is a sensible choice.
Yes, exactly. Because for the prediction model, especially for like say if you want to communicate with clinicians, like even though we have a very good model, like they can predict the patient's outcomes, it would be very hard for them to use and to interpret.
Yeah, so another question is like, and that actually applies also for the previous project. So like, the main concern when you work with real world data would be confounding, for instance, or violation of positivity in the previous project. So if you are expanding the eligibility criteria, but no one in this under this new eligibility criteria has gotten the treatment, or in this case, if the model that you are fitting is there is like hidden confounding, like because you are looking at genes, maybe that's less of an issue, but like more generally, like is like how would you go about hidden confounding and violation of positivity in projects 1 and 2?
Yeah, that's a very good question. So yeah, I think like say one of the largest difficulty, or like say the point we should take care of, is the confounding like like the difference we have in this real world data in medical records. So currently how we do this is say we run our model, like say our model considers or like already adjusted or control for those confounders. And we do have some statistics metrics to evaluate like the imbalancement or like the how well you adjust for it. Like you can say there are different statistical metrics to test. And apart from that, so that's also why for both projects, we try to validate it on some of the clinical trials data. Because say on the clinical trials data, you no longer have the selection bias because all the patients are randomly assigned. So that's also why, for example, I showed this case study, like say we try to aggregate some clinical trials to do some of this analysis. So basically it's 2 steps. First is like say you can to the best as we do as best as we can to adjust for those and have a different metrics to test how well we do this adjustments and control. And second is we validate subset of our results on clinical trials data. Yeah. So basically like repeat our analysis on clinical trials data. And also because also like the significance of like the real world work site like this is because you do not really have so many clinical trials data to test for, and also like so with genomics profile, for example. So basically we can only validate a very small proportion of our results on clinical trials because we do not really, it's also very costly, like you do not really have those. So that's also the power of the real world analysis.
Thanks.
Yeah, I think I agree. Like the best practice was still to run clinical trials because like from like we can do whatever we can from the methods, but like still there can be unmeasured confounders, etc. So that's also why we tend to frame this as a hypothesis generation. So we propose those could be good candidates we found from real world data, and there could be good candidates for, say, for example, in future, like if you build clinical trials or you want to identify good biomarkers, those could be good candidates still. So this would not be a hard conclusion. It would be just hypothesis we get from the real world data. Great. So maybe I can quickly conclude saying like in this talk, I mean presents like say both how we can use real world data medical records combined with machine learning tools to help clinical trials design and also to generate new hypotheses for precision oncology. So I would acknowledge all our fantastic collaborators who work together in these 2 projects. So my advisor, James, and also all our collaborators from Roche Genotech. Yeah, thanks for attention. So yeah, I love to take a question for both of those 2 projects.
Thank you so much, Ruishan. That was an amazing talk. Any questions from the audience? Yeah, I guess one question I had before, like maybe someone else thinks of other questions, is like uncertainty quantification. So in your project 1, like you are optimizing the selection criteria based on Shapley values, but you have like point estimates of the hazard ratio under different criteria. Like, would it be of any value if you also have confidence intervals on these hazard ratios, and like can you still use the Shapley value optimization if you have like interval estimates of hazard ratio rather than point estimates? Because probably you'll get less uncertainty as you explore different criteria than what.
Yes. So I think you raise a very good point. So currently we just give a 1-point estimate, like and also like basically also why we use Shapley analysis is more interpretable, like say give one evaluation for individual rules. So I think like what you raised is actually say our future directions we want to add into this We also have another module like a factorial optimization, where like is to say give a best set of rules would give you the best hazard ratio. So yeah, I think like also what you raise is also another module, like would be a very good module we want to add to the tool. Yeah.
Other questions, anyone?